Hello pre-AP biology students. Welcome to this video talking about the concepts of meiosis and mitosis. Today we'll be focusing on how it works, the cell division, its importance, and many other factors, and especially how seen in real life. This is a slideshow and also a video, a concept to you guys explained by AP students right now. Me, your presenter Lewis, and Prince, your scripter. Please don't give up on this video because it's definitely worth it. It's a hidden gem. It's something to watch. So bear with me here as much as it's really annoying and hard to understand this concept. First, we start with cell cycle. The cell cycle is an important part, so because it's necessary to understand it in order to see how cell division works. Cells that replicate in for three main reasons, being reproduction, growth, and repairs. All cell division starts with the cell cycle, so it's the first step being the interphase. The cell cycle has four parts, but three are the most important, being G1, S, G2, and G0. G0 is more of a resting phase of the cell, and it's when the cell has already finished replication or it is just not preparing itself at all and it is not necessary for it to replicate. G1, known as growth phase, is the phase where the cell starts to grow. The cell grows in size, it creates more organelles and proteins, being known as the cell startup phase after division. It allows the cell to check if the conditions are right to continue on to the next stages. The next one is your S phase, the synthesis phase, is DNA synthesis. It replicates the DNA in the cells, making a complete copy of the genetic material. Each chromosome is duplicated, then forming sister chromatids. All this is made to ensure that each new cell that is created from this process then gets the same DNA it needs. G2 phase is the second growth phase of the cell. The cell continues its growth and now it begins its preparation for meiosis or mitosis, producing proteins and organelles needed for the division, like spindle fibers. A really important thing that's going to be seen in the next sort of phases of mitosis or meiosis. With this, the cell just double checks again that the DNA replicated is complete and successful for then entering one of these the next stages, whether it is meiosis or mitosis. Understand that this is one of the more kind of tedious ones to understand, just to kind of memorize that this isn't all separate, but it's all in the same step of interface. There's a lot of questions that come up with how mitosis and meiosis works and what it even is and how it's formed. Mitosis, shown here as an example, is one of the types of cell division. It produces two genetically identical daughter cells from a single parent cell, which mostly happens in somatic or body cells. It can be seen as an essential need for growth, tissue repair, and maintenance of an organism in general. But way before all this can happen, of course, as I explained, mitosis undergoes a phase called interphase. Interphase is the cell cycle, which was G0, G1, S, and G2 phases. All these phases ensure that the cell will receive an identical set of chromosomes to the parent cell. So it's highly needed and it's highly seen, but it's kind of in both do the exact same in both interfaces when shown in mitosis and meiosis do the exact same thing in both sections, but it's important to understand that they are a crucial step and are needed to happen. The entire process of mitosis after this includes of prophase, prometaphase, metaphase, anaphase, and telophase followed by cytokinesis. Prometaphase isn't shown in this example because it's not there, but it is something crucial in one of the steps. It's after all this, it results into two diploid cells that are genetically identical to the original cell. This is a crucial thing in multicellular organisms due to it being used for growth, development, and its ability to increase the number of cells needed for survival and healing. Additionally, some organisms use mitosis for asexual reproduction, producing offspring genetically identical to the parent or organism. Meiosis, as shown as an example right here, is a specialized type of cell division that produces 4 genetically unique haploid gametes, or sex cells, from a single diploid parent cell. It occurs in germ cells and is essential for sexual reproduction and genetic variation. Meiosis consists of two different kinds of divisions, known as meiosis 1 and meiosis 2, during which the first one will be a homologous chromosome and followed by the second division being a sister chromatid. The process reduces the chromosomes numbers by half compared to what mitosis does. Meiosis increases the genetic diversity through crossing over and independent assortment. Processes that create unique combinations of genes and make each resulting gamete genetically different from the parent cell and from each other. The importance of this genetic variation is how critical it is for evolution, adaptation, and survival of many species. It is seen in a lot of the world right now. Many animals are forced to mutate and combine with each other, thus going through meiosis as they reproduce. It creates variation. It allows people to make more different kind of species in itself. Without meiosis itself, we wouldn't even be here since we are technically evolutions of many species. Mitosis has many phases in its code. It has 6 phases that we see. One already explained, interphase, which is G0, G1, as in G2. And then we undergo prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. It is kind of tedious to understand it. It is kind of long, it looks long, but once you get the understanding of it, it's kind of basic. Prophase is the first stage of mitosis, in which the cell begins preparing the genetic material for division. In this stage, chromatin, which is loosely packed DNA, begins to condense in distinct divisible chromosomes, each consisting of 2 identical sister chromatids joined at the center. The nucleus disappears and the nuclear envelope starts breaking down, allowing the cell's genetic material to become accessible for separation. Centrosomes then move towards opposite poles of the cell and begin organizing into spindle fibers made out of microtubules. These spindle fibers are essential parts in the later stages of mitosis, since it then attaches to chromosomes and guides their movements. You can see how it works right there in the prophase. And we move on to prometaphase. Prometaphase is following after prophase, is characterized by the complete breakdown of the nuclear envelope, allowing spindle fibers to then enter the nuclear region and attach itself onto chromosomes as specialized protein structures called kinetochores. These kinetochores are located at the centromeres. Each each sister chromatid becomes attached to spindle fibers originating from opposite poles of the cell, ensuring that they will be pulled apart correctly later on. During this process, chromosomes also begin moving towards the center of the cell as spindle fibers actively pull and adjust their positions, and uh, and this is more or less seen as a critical stage. It establishes physical connections needed for accurate chromosome separation later on. Metaphase, following after what prometaphase has begun, allows for the chromosomes to align along the center of the cell, an imaginary plane known as the metaphase plate. So you can see right there, that's the metaphase plate that's written down. Each chromosome is fully attached to the spindle fibers from both poles, creating a balanced tension that ensures proper alignment between all chromosomes. Being named the spindle checkpoint, preventing the cell from proceeding until all chromosomes are correctly attached. This stage is essential for ensuring that each daughter cell receives an identical set of chromosomes, as the precise alignment guarantees equal distribution. Anaphase is what follows after metaphase, and this stage is one of the most critical steps of mitosis itself. It ensures that, in general, that the future daughter cell will receive an identical copy of a genetic information that the parent cell was duplicating in general. The sister chromatids are fully separated at the centromere and pulled apart toward opposite poles of the cell. Once separated, each chromatid is now considered an individual chromosome. The spindle fibers shorten as they pull them in, while the cell starts to elongate itself to help increase the distance between the two sets of genetic material. Telophase. Telophase is now practically the reverse version of prophase. Once the chromosomes reach the opposite ends of the cell, they begin to decondense back into less visible chromatin. New nuclear envelopes form around each set of chromosomes, resulting in the formation of two separate nuclei within a single cell. The spindle fibers disassemble as they are no longer needed, and from this point, the cell has successfully divided into genetic material equally and is preparing to physically divide itself. Cytokinesis is the final and last step of this of mitosis by dividing the cytoplasm into two separate daughter cells, and it's a little bit different between animals and plants. In animal cells or mammals, it occurs through the formation of a cleavage furrow, pinching the cell membrane inward until it splits. Unlike plant cells, where a cell plate forms in the center of the cell and eventually develops into a new cell wall. The end result of this stage, at the end, both, no matter which one it is, will always end up into two genetically identical diploid daughter cells with the same amount of chromosomes as the original parent cell. As long as this isn't tedious as it is a really understanding and easy part of life that is seen everywhere, and examples come everywhere around you. You can even see it in yourself. A great example of mitosis and how it works can be somebody getting a cut on their finger. As you can see that example, it demonstrates the wonders of how your body repairs itself from a cut or most kind of injuries. It's through mitosis. The healing process includes the damaged skin cells needing to be replaced through controlled cell division in order to restore the lost tissue. When an injury like a cut happens in the body, neighboring somatic cells are stimulated and enter the cell cycle and undergo mitosis, producing identical diploid daughter cells. Of course, it isn't only a single copy, it's thousands or even millions, depending on how big the injury is. Allowing the tissue to regenerate and finally close the wound. Because of how mitosis itself preserves the chromosome number and maintains identical genetic information in each daughter cell, the repaired skin is able to maintain regular structure and function, and you'll basically just heal. That's seen as you get a cut, it heals back to normal. You gain function, and you're back to normal. The process is something that is seen in day-to-day life that demonstrates how mitosis plays an essential role in growth, tissue repair, and cellular maintenance within multicellular organisms like us. We grow only because of mitosis. We end up growing, we get taller. It's all mitosis work. But mitosis can't compare to what makes people and multicellular organisms so unique. That job alone is taken by meiosis. Meiosis is what mitosis can't do. It's what makes people so unique and allows people to be so different, even if you were to have the same parents. Meiosis undergoes two separation phases, compared to mitosis only being one. As seen in the chart, meiosis 1 and meiosis 2. It's similar, but it's still different. It's all different phases. It is a lot more longer, a lot more tedious. It's a lot more annoying to understand because it's longer. It's more stages, more phases, more steps. Prophase 1 is the longest and the most complex stage of meiosis because of many critical events responsible for genetic variation, which occurs during the phase itself. Chromatin condenses into visible chromosomes, each consisting of two sister chromatids joined together at the centromere. Homologous chromosomes pair together in a process called symphysis, forming structures known as tetradrads. While this happens, crossing over is also happening, where homologous chromosomes exchange segments of genetic material, creating new combinations of alleles, allowing for genetic variation to flow. As this all is happening, it follows similar steps to how mitosis prophase works. As a nuclear envelope begins to break down, the nucleus disappears, and the spindle fibers form to as centrosomes move toward opposite poles of the cell, preparing for the separation of homologous chromosomes. Metaphase 1, the homologous chromosome pairs or tetrads align along the metaphase plate at the center of the cell. Spindle fibers attached to the kinetures of each homologous chromosome from opposite poles. The orientation of each chromosome pair is set at a random, a process called independent assortment, which is another way that meiosis creates genetic variation. It contributes greatly to creating different combinations of maternal and paternal chromosomes in gametes, connecting to one of the many different differences meiosis and mitosis have, since chromosome pairs align together during this phase. In anaphase 1, homologous chromosomes are now separated and being pulled toward opposite poles of the cell by spindle fibers. The sister chromatids, however, remain attached at their centrosome and do not separate during this phase itself. The separation of homologous chromosomes reduced the chromosome number by half, making meiosis a reduction in division. The movement of chromosomes during anaphase 1 ensures that each future daughter cell receives only one chromosome from each homologous pair. Telophase 1, the second to last step to meiosis 1, has the homologous chromosome arrive at opposite poles of the cell. In some organisms, the nuclear envelope may briefly reform around the chromosomes, while in others they might not fully reappear. Cytokinesis then divides the cytoplasm, producing two haploid daughter cells. They Even though they are haploid, each chromosome still consists of two sister chromatids at this point. At this point, the chromosome number has been reduced, but the genetic material is still duplicated. Meiosis 2 consists of the same, just that they aren't one but not two. Prophase 2, metaphase 2, anaphase 2, and telophase 2, and cytokinesis. Though like they have really similar names, they are different between. There are many differences between each other and how they actually function and what they do. As meiosis 2 uses two haploid cells that were produced from during meiosis 1 replication to then make four haploid cells. Prophase 2 starts off in the two haploid cells that were already produced by meiosis 1. It doesn't matter which one I mentioned because both of them are still doing the exact same thing. So chromosomes still consisting of sister chromatids condense again if they had previously decondensed. The nuclear envelope breaks down if they had reformed, and new spindle fibers develop as the centromeres move toward opposite poles. The biggest difference between prophase 1 and 2 is how there is no crossing over or synthesis to occurs because the homologous chromosomes have already been separated at this point. There's no point for more variation. Metaphase 2, the chromosomes align individually along the metaphase plate of each cell. Spindle fibers attach the kinetures of sister chromatids from opposite poles. The arrangement is similar to metaphase and mitosis, but the cells are not haploid cells compared to diploid. Rather than this also ensures proper alignment, and the sister chromatids will now separate evenly during the next phase that's coming, which is anaphase 2. Anaphase 2 has the centromes divide, and the sister chromatids are finally separated by spindle fibers, pulling them apart into opposite poles of the cell. Once separated, each chromatid is considered an individual chromosome. This stage ensures that each resulting gamete receives a complete set of chromosomes. Telophase 2 chromosomes will finally arrive at opposite poles and begin to decondense back into chromatids. Nuclear envelopes reform around each set of chromosomes, and spindle fibers disassemble. Cytokinesis then divides the cytoplasm in each cell, resulting in the formation of four genetically unique haploid daughter cells. These cells function as as gametes containing half the chromosome number of the original parent cell, making them suitable for sexual reproduction. This is a really important step. It's seen in day-to-day life, and it's so many more examples in the real world, as you can see. Um, and it really works for even us as people on how we look, how we see things, how we do things, even the way we're just. If you have siblings at home, or you have uncles and aunts, people you're related to, you can see, or even friends if you're like a single kid. Look at their siblings. Look at how they look alike. Look at your brother, or sister, or even any kind of family that has siblings in general. You ever wonder how even though we're born from the same parents, we still look different? This is the greatest example of how meiosis works in the real world. You and your sibling will almost never be during uh, genetically identical due to meiosis. The process is like crossing over and independent assortment, shuffle genetic information, creating unique combinations of genes in sperm and egg cells. This being said and known, it directly dictates to why siblings may share similar traits like eye color or facial shapes, or even facial features, but still have different heights, personalities, hair textures, and etc. The genetic variation created by meiosis is extremely important because it creates diversity within a species and helps the population adapt and evolve over time. Take this for an example right here for two siblings who are born from the same parents but still look different. Also, here another example right here is example. Let's say these two. The origin of the photo isn't too important, but let's say these two dogs are from the same litter. The one is a complete menace, while the other one is afraid of it. Also, one has black fur and the other one has white fur. But their actions and the way they act is what makes them different from each other. Your sister can be a complete nutcase while you're a normal child, but you know what? You both still share the exact same genetics, so you just gotta run with it. There are so many differences between meiosis and mitosis itself, and we come to find out what the differences are, why they're so different, and more of a biology look into it rather than a real mitosis and meiosis has a lot of differences that are seen in day-to-day life. These are the most basic differences in itself, it being how the type of reproduction, the kind of cells, and the altered number of chromosomes, and the count of cell division cycles. Mitosis, of course, only has a single cell cycle, cell division cycle, which is a single one, and meiosis has two. While as in mitosis can be used for asexual reproduction and only asexual, while meiosis is used for sexual and only sexual. Mitosis creates two daughter cells, while meiosis creates four. The mitosis creates cells that have the same amount of chromosomes as it, but meiosis creates ones that have half. There are a lot more examples, as I can explain right now, how it occurs for meiosis has it occurring in germ or reproductive cells, while mitosis has it occur in somatic cells. While mitosis is used for growth, repair, and maintenance, making it an important factor for healing and tissue repair, meiosis is used for sexual reproduction, important for genetic diversity and evolution of a species. Mitosis doesn't cross over because it doesn't make different kind of things. It makes everything identical. While in meiosis has crossing over and independent assortment. The biggest thing to pull out this is that mitosis is more of a duplicate. You want to make something identical to yourself, while meiosis wants you to make something completely unique. It is similar, but it's unique. It's like when you're designing something, when you take an example and design something new off of it, you make something uniquely new. Thanks for sticking around and watching me talk about meiosis and mitosis, and also thank you for deciding to stay here for me, your presenter this, and of course the scripter who made this possible, Prince. It's an interesting matter. This is not only seen in school hours, but it can be seen everywhere around the world, especially with your own body. thank you for watching this, everybody, for incoming pre-AP biology students. I hope you enjoyed it and have a good day.